So, good morning or good afternoon, everyone. So today I'm going to be presenting one of the projects that we have going on in my lab here. And for those of you all that don't know me or haven't, met me before, so my name is Robert Stewart. I'm in the Department of Mechanical and Aerospace Engineering. And the research in our lab focuses on cellular biomechanics, and cellular biomechanics is essentially a blend of physics and cell biology. So we strictly do cell work within our lab. And the idea is to really link mechanics to human health and physiology. A lot of the major projects we have are cardiovascular related, but we also have some projects related to diabetes as well as in the neurosciences as well here. And our lab is in Lake Nona as well on the health sciences campus. All right, so what I like to always start off is just first acknowledging the funding sources and the students who really made this research possible here, because without them, we, as professors wouldn't have anything to talk about. But the funding sources, this project was funded by NSF, NIH, all of my lab members, and Vignesh, who is a former PhD student in my lab, who's currently a postdoc, is the student who's responsible for the work, actually most of the work that I actually be presenting today here. So before I go into the project, I'll just kind of quickly talk about the main methods that this project is based off of and what it's all about. So you'll hear a lot of talk today about, cell-derived biomechanical forces, tractions, and intercellular stresses. So kind of a quick run through of what do I mean when I say that. So essentially the tractions are going to be those mechanical stresses generated between the cell and its substrate. Okay, as I have here in the arrow, we use a method called traction force microscopy to calculate those stresses. Now, the key to this is that we have to have cells physically attached to a surface, but this surface has to be flexible, because what happens is we actually track the displacements within this surface, and then we calculate what the forces are and subsequently the stresses exerted by cells on the surface of this flexible gel here. We also use another method called monolayer stress microscopy to calculate the intercellular stresses. Simply put, these are going to be the mechanical stresses generated between cells. A note here, we're talking about between cells, and we're not looking at the actual stresses within cells here. So that's just a FYI. And when we, part of what we can generate when we calculate our intercellular stresses is the normal stress and the shear stress, right? And if we look at that little image at the top right corner here, you can see where those are oriented relative to a cell junction here. And some of you all who've seen my talks have probably seen this slide, this part before, but I always like to describe the interaction as, people, if you're playing a game of tug of war, tractions is going to be, we look at these guys here, tractions are going to be that, so stress is generated as they're bracing themselves to push off the ground. Intercellular stresses, that's going to be the tension in the rope here. Okay, so I'll first start off with, what is the extracellular matrix here? Kind of a quick run through of that. So it is a family of proteins, and it's a family of scaffolding proteins. Some of the ones I have here are more of the common ones that exist within the body, and there are even subfamilies within these, like collagen. I think there are about 30 different isoforms or structural configurations of collagen. Same for fibronectin and laminin. Okay, it is a supporting scaffolding that cells physically attach to, and that's whether they're anchorage dependent or not. Okay, it's a dynamic structure, right? So yes, cells are physically attached to it, but the cell and the body in general can dynamically rearrange this physical structure here. But I always like to point out that beyond just being something that the cell is physically attached to, it is believed, and there's evidence to show that it functions as a biochemical signaling reservoir. And what does that mean? So essentially that means that you can take the same cell type, put it on two different types of extracellular matrix proteins, and you'll get different responses biochemically. You can get differences in protein expression here. And it's certainly been, since cells needed to attach to, certainly it has been shown to be important in a lot of mechanics related processes, such as I have here, migration, adhesion, etc. Even important in inflammatory response. Certainly important for blood vessels, as I have here, and important for just basic physiology and pathology here. So that's a run through of what the extracellular matrix is, and hopefully, there's an understanding of that. But let's go to the why, right? Well, why should we care? Okay. And I always like to say, not all ECMs are created equal. Okay. And what does that mean? Well, depending on the tissue type, even within the different blood vessels, you will get a different extracellular matrix composition. In the previous slide, I gave you an example of about 3, right? So is it usually just going to be one specific protein within the blood vessel, within the tissue? No, it's going to vary depending on the function. Some ECM proteins will make structures more stiffer, some will allow them to be more soft, okay? So the main point here is that you'll get differences based off of where tissue is located physiologically, but also pathology plays a role as well, okay? As an example, as I have here, we take our arteries, okay? These are the major blood vessels within the heart. These are the blood vessels carrying blood from the heart to be delivered everywhere within our body, right, to oxygenate our tissues, right? It's going to consist of fibronectin, laminin, and collagen, right? Collagen is going to be the primary extracellular matrix molecule, and it's actually type 1 and 3 primarily consist of that. But again, we have fibronectin and laminin as well here. But as I also have, right, that composition, literally that chemical structure will change as a response of various pathologies. For example, it's certainly been well established that diabetics, their blood vessels are going to be more rich in fibronectin and actually stiffer as well for a host of reasons. Individuals who have atherosclerosis, again, you will see this shift in the primary composition of the ECM from less collagen to more fibronectin rich. Okay, so the main point is that whether it's a physiological implication or pathological implication, we see a difference in the ECM composition. For those of us that do cell culture, generally what happens in our labs, we have our favorite type of extracellular matrix molecule that we use and we stick with it. But the idea is, well, how would that affect the mechanics, right? Because you want to be consistent in your coatings, but is there an influence at all here? Okay, so what's the link is the why. So the how, to kind of quickly go through that here. Oops. So the how, we use HUVEX, human umbilical vein endothelial cells, cultured them on hydrogels. And what we did was we varied the ratio of various ECM proteins. Now we use collagen and fibronectin, and essentially, as you see here, we started with the 100% collagen, and we just varied it where we went from more, complete collagen, adding fibronectin to where that ratio was similar. And then we, slowly decreased it to where that matrix was 100% fibronectin. It should be noted that these were all independent experiments, okay? So coated the cells, different ECM compositions. And we did, our basic mechanics measurements. So we looked at the cell-derived mechanical stresses, tractions, and intracellular stresses I mentioned earlier. We looked at kinematics, so cell velocity, right? How fast are the cells moving, right? Cell morphology, does the shape change, right? So the idea is, we're simply just changing that protein that the cells attach to, and we want to see the influence it has on the mechanics, okay? So going into the results here, so the phase, this is just showing morphology. The cells look, pretty similar here, nothing at least, from the eye that looks distinguishable. Looking at the traction, so, I'll say that this behavior that we saw was a bit surprising, but what we see is that we go from the 100% collagen all the way to the, we get this maximal response or maximal magnitude of the tractions when we have this 50-50. But if you look at the end, when we completely culture the cells, on 100% fibronectin versus collagen, we see that they actually almost look the same here. And this was a behavior that we actually saw, for our other measurements, where during this transition, and you certainly get this increase in mechanical stresses, but we see that as we're going down, switching to another matrix, it kind of equilibrates. So the importance here is that this transitioning is actually, what happens in the body. You never have a 100% collagen, 100% fibronectin, anywhere within the body, actually. So we'll look at intracellular stresses, similar behavior here, where we're going up maximum response at the 150-50 collagen fibronectin here. And then we see, I'm not sure what happened with this 25%, 75%, but nonetheless, this is what the data showed. But again, see this adaptation here. Okay, so intracellular stresses, cell area again showed the same response here. Again, so the main point is that we are certainly getting a change in just how the cell behaves mechanically, physically, by simply just modifying that ECM protein here. And more importantly, of course, is that it's the ratios that actually matter. And we only use 2, but of course, as I talked about earlier here, the native ECM is actually consists of multiple proteins at any given point in time here. Interestingly, the velocity was a little bit different. So we start with our 100% collagen. We actually see a decrease. So the cells are moving, they appear to actually move slower when we start to actually, manipulate these ratios. We switch to the 100% fibronectin. It actually, that velocity actually hasn't completely recovered, relative to the collagen, but nonetheless, a bit of an opposite response here that we saw. So in summary, to kind of, conclude everything, the main point, right, of the take-home message, as I have here, right, that the why should we care, whether the ECM composition certainly matters, and certainly that ratio will influence how the cells will actually behave mechanically. And we were able to influence that magnitude just based off of the concentration. I would say future works would certainly, that we would want to look at is actually trying to generate, a matrix composition that's more, I'll say, physiologically relevant to blood vessels, since that's the system that we work with. But the idea is, again, in our labs, anyone that does work, we all, have a protein and we stick with it. Some labs like fibronectin, some labs like collagen, and, that's all that, we work with. But the idea is that you will get a response. What I will say is whatever ECM you're using, certainly don't switch in the middle of an experiment, because you will get a change here, right? And we looked at the mechanics. Certainly, the idea is we like to blend these mechanical responses with protein expression, because there are some proteins that we know are related to the generation of mechanical stresses and mechanical forces. So really teasing out more the role that this protein plays is something that we're going to look at for future work here. But with that being said, that is the end of my discussion, and I'll take any questions that you all have here.